Swayam Prabha. Digital India. Educated India.
Okay, students, hope you are very well. So today I will start the first class on nanobiotechnology and our point of care. So mainly today I will cover the basic introductions part on nanobiotechnology and our point of care devices. So why this technology is very important for the disease diagnosis, and in also in the current scenario why this technology is really important for you, that I will cover first. So let us come to the syllabus or the topics that I will cover during the whole courses. So mainly I will cover in the introductions of the basics point of care diagnosis, then its analytical parameters for biological applications, and then I will cover all the diagnostic aspects and how all the biological molecules and nanomaterial they are interact and they are used for the diagnosis, that I will also cover here. Then also I will cover the nano materials and their fundamental concept, their synthesis, how they can be useful, they can be used for diagnosis, and then I will cover the basic tools for the diagnosis. Basic tools means the technology, the technique that we will use for detections of the sensors, detections of the biomolecules, that I will cover here. Also next I will cover the signal amplifications. Signal amplification really is a very important part for this nanobiosensor this course, because you know the main concept for this course is the early diagnosis. So if you want to detect the disease at the very early level, then signal amplification is really important, because suppose you have some disease marker in our some blood samples or in the saliva sample, on the urine sample, very low level, and it can cause the disease, although in the very early condition we should detect them. But if you cannot amplify the signal, because and if your device cannot detect at the very low level, then it is very difficult to go for some decisions at the very beginning. So suppose for the cancer, if you cannot detect the early stage and if you detect the end stage, so although we know that yes, this marker is present, but as it is a very high concentration, doctor may prescribe go for therapeutic process, but it is difficult to save the patient life at that time. So detections of the disease at early stage is really important, that we will focus mainly here. How we can develop a sensor device that can detect at the very low level. So that is called ultra sensitive way, or means ultra sensitive biosensor will develop. So that's why signal amplification is really important. And for how we can amplify, different kind of chemistry we will employ here. That is called signal to background ratio. So that I will describe what is the signal, what is the background. So that we will describe, and we will use some redox cycling. You can see here this redox cycling technology method. So this is really important for the signal amplification. So here we will use some amplification technology based on some chemistry. Also we need some electrode surface modifications with some nanomaterial, because in this course nanomaterial will be used always for surface modification. As I mentioned that synthesis of different kind of nanoparticle is really important, that all the technology I will teach you in this course. It is very important part of this course. Then how this nanomaterial can be used for the device development, that is also very important part, because nanomaterial has a unique property, they have very high surface coverage. That surface coverage can be useful for different biomolecule immobilization on the sensor surface, and this can help for increase the conductivity, increase the surface area. We can easily miniaturize the sensor, that is why use the nanomaterial is very important. Then I will come the lab on a chip devices. So what is the lab on a chip? So that also I will describe. Lab on a chip concept is very useful for miniaturize of any device. Generally you can see in the lab we are using very sophisticated instruments, very large instrument, but it is very difficult to use those instruments for point of care. So, what is point of care? Point of care means near the patient you are using your diagnostic method. Suppose in the lab we need a very bulky instrument, that is very expensive, but especially for the developing countries use this kind of the very expensive method or protocol is really difficult. So, point of care means at the same time we have to think about the cost effectiveness. So, it should be near to the patient, at the same time cost effectiveness, that also we will cover in the lab on a chip. Mean all the lab concept will bring to a very small chip, and that chip can be used near the patient. So, I will give you some example, I will show you some device kind lab on a chip things. Then multiplex diagnostics I will cover. This is very important part of the multiplex diagnostics, because see, one patient may come to the doctor, so he has some disease, like maybe some conditions doctor can find it, yes it has some disease, but there is some other disease also can affect in his or her health conditions. So, not only the one disease marker, at the same time we should detect the multiple disease marker to take the a final decision. So, doctors can only take the decisions if he can detect the multiple analyte. So, like at the rainy seasons you come to a doctor with some fever. So, it may be bacterial or it may be viral fevers or it may be something else. So, doctor should detect not only the bacterial, it should detect the something viral, also maybe it's dengue or maybe malaria, right. So, which one we do not know. Also, if has some other cause, maybe during this COVID time. So, maybe due to the COVID. So, we should go for the COVID diagnosis also. So, that is why at the same sample we can detect the multiple disease at the same time, that is called the multiplex diagnosis. That is very important to the final decision to a doctors. Then I will cover different kind of the non-invasive, invasive approach, the detection of the glucose. So, glucose generally we are detecting the pricking the finger, taking the just drop of the blood and putting in the on a chip. But this pricking the finger is called the invasive because it is painful. We can go for some non-invasive approach also. Non-invasive means without pricking any finger, without taking the blood samples like saliva samples, urine samples, sweat samples, they are the non-invasive approach. Those technique also we can use. So, this all the techniques, how this kind of the samples we can use. So, then you can ask me the questions that why then people like the blood samples. Yes, the blood sample is a very good sample, that because this is directly we can get so many analyte in the blood, and their concentrations is very much related. I mean, you can easily predict the disease level, but there is always some correlations with your saliva biomarker with your blood biomarker, or maybe sweat biomarker with your blood biomarker. There is some correlations already present. So, that is why even blood is a good sample, but still we can use the sweat sample, saliva sample, urine sample. There is some correlations that I will describe during the class, and that non-invasive approach will be very beneficial, be useful for point of sight applications, because no need to we need some specialized person to take the blood, but common people for point of care diagnosis they can take the saliva, urine sample, they can easily collect. So, sample collection is not a big issue. That is why non-invasive approach is very important part for point of care device development. Okay. So, then we will come to the printed bioelectronics. So, why the printed bioelectronics is really important for the point of care, because we can use like a banded kind of things like flexible printed sensor. You can print the sensors even on your skin. Some patch kind of the sensor we can develop that can continuously it can monitor, like whole day it can monitor the biomarker level like on your sweat sample. So, that is why printed sensor also very much useful for the point of care device development, that I will cover also. Then some new detection technique, although so many detection techniques already available in the market, but still we need some new detection technique, because you see during the COVID time like some new disease may come and how easily we can detect them, means how patient easily can handle them. Many technique is already available, but in the market we can see very less number of the device available. So, why? Because there is some difficulty people are facing when they are commercializing their device. So, we need anyway some protocol or some technique that is very useful for end user. So, end user means like you may like we can use in the like in our home, like just take the saliva sample and test it within a few minutes, you see the level of biomarker. Even we can use your smartphone, we can see the value like digital value, this much biomarker present, right. So, this is new detection protocol we need, and we can easily so that this protocol or this technique can easily integrate with our mobile device. See, like everyone has the mobile phone. So, if we can easily integrate our sensor, like there is a you can see here is some always like charging point. So, if we can like I can show you some chip. So, okay, see we can develop like this kind of printed chip. So, see there is a connector. See, in your smartphone here is a charging point, if you can easily see connect here like this, then we can develop some app here in the smartphone. Now, with this app we can control like your chip, and here on the chip in the sensor surface, now you can drop some sample here and there will be some chemical, and sample chemical will start reactions and it will generate some electron. That electron will transfer through this chip, and easily we can detect and we can show on your device skin this digital value we can see. So, that it is very miniaturized, very small device. So, this kind of electrode, this is called the electrode, printed electrode, we can easily develop, we can use for the disease diagnosis. See, this is very small and it can be easily means very miniaturized, and near patient it can be handled easily. This is one simple technique that I will cover during the whole class. So, next part I will cover mainly device commercialization. See, device commercializations, as I said, there is many technique available, you can see lots of publications, but why all the technique is not in the market? There is certain challenges exist, right. So, that challenges I will address, and then also I will address how we can overcome those challenges and we can bring those technique in the market. That is called commercialization possible, how we can easily you can bring to the market. That is really important part of this. So, this course, that is why it is not only the theoretical. So, I will teach you some practical based some lab class also I will take at the end of this course, and I will tell you how easily you can fabricate all the biological part and the nanomaterial part on the sensor surface, and then a miniaturized system you can develop, and how these systems you can easily bring to the market. Because some of you are really you want to start your career, like you want to start some startup or something your own company, right. So, this is a really important part in the biomedical device development. So, this kind of new technology you can think, and you can simple way you can develop some device, and if it is really useful for end user you can bring to the market. Just for one example like glucometer. So, it is see huge market it has, because it is very simple method, and just drop of the blood and you can detect the glucose. I think nowadays everyone every home they have one glucometer. Because it is now not that much expensive, at the same time it is user friendly. So, similar kind of device if you can develop and if you can easily commercialize, it will really impact not only for your own career, at the same time it is good for your society, right. You develop some technique that is useful for common people. And again I will discuss some biosensor device that can be useful for environmental monitoring. So, not only the disease, this kind of device can be useful for on-site for field testing, like health can be our health. There is a different kind of issue can be possible because of water contaminations, right, like arsenic, lead, this kind of heavy metal also can contaminate the water, it can also impact our health. So, this is a health related, right. If you can measure the arsenic level, lead, this kind of level in drinking water or even in the soil, that will be huge impact, right. So, that is why we can I will show you some sensor development that can be useful for also at the same time environmental monitoring that can be useful for field testing also. Also the food safety. So, we are taking the food that food may not be in the good conditions, right. So, how we can determine the food quality? That is also directly related to your health. So, we have to determine the food quality, that sensor also we can develop. And finally, security means you know, like bioterrorism can happen anytime. Also some some molecules maybe in your environment or during the terrorism attack, maybe some biomolecules they can spread and that can affect on your health. So, this kind of biomolecules also different molecules we can detect from different samples, even in the water or some other samples also you can collect, and we can use this one for security purpose. Even in the alcohol detections, that is also useful for different security purpose we can check. That I will cover. Okay. Now, come to the main keywords for this basic course that I will cover. So, main is the nanobiosensors, means why is the nanobiosensor? So, what is the biosensors that I will teach you today, and why it is called nanobiosensor, that I am going to tell you in the next slide. Now, point of care. So, point of care means the near the patients, how you can use a technology that you are developing in your lab, near that technology can be useful for patient, near the patient. This is something called the bench to bedside applications. So, point of care application sometimes it's called bench to bedside. Bench means in the lab what you are developing, that you can bring to the near the patients. Then lab on a chip, this is a very fundamental and very important part for this course, means all the lab technology you will bring on a single chip. Like I told you, like single chip, just I showed you this kind of single chip. So, there you can bring. Then smartphone based sensor, like this kind of chip you can easily integrate with your smartphone, because many people now everyone has the smartphone, right. So, how we can easily, if you can develop some sensor, if you can easily integrate those sensor with your smartphone, then easily you can use. That is why smartphone based platform is very important. Then finally, commercialization of the device. Okay, let us start the current medical conditions. This is the very important part of this course. This is a very basic introductions. Now, I am going to tell you that what is the current medical conditions and its diagnosis. So, you know, there are many kind of disease now, like HIV, malaria, tuberculosis. This is the very common disease nowadays. And recently, I think you know, like the SARS-CoV-2, this COVID-19, these times we really want to detect like this kind of virus, and then this if you can detect this kind of virus very easily for point of care setting, it is really important and it will be very useful, so that we can stop the spreading this SARS-CoV-2, right. So, if you can yourself, sometimes people actually worry to go to the hospital and go for the diagnosis for SARS-CoV-2, right. But if you have their own like device in their home, they can easily detect, and they can tell their family member, yes, I am affected. So, I can keep myself in the quarantine in a separate place, so that this virus will not be spread among our family member at least among my society. So, this kind of some decision you can take, right. And different chronic disease like diabetes, obesity is very common nowadays. So, this kind of medical condition, if you see, like almost in our if you think about India also, I think people are almost many people they are affected in this diagnosis, this kind of disease, and we need some certain diagnosis, so that at the very early stage we can take some decisions. But what is the problem in this current stage? If we go for, and if you ask the doctor, sir, I want to go for this diagnosis all the disease, then they will collect the sample. What sample? Mainly they are collecting the blood, and then they will send to the laboratory, mainly laboratory based, like this kind of like very highly expensive and big instruments they are using, right. And this diagnosis protocol or method is very laborious, because first they will process your blood sample, they will separate the blood cells, they will collect the serum, then from this serum sample they need sometimes the pre-processing. For this pre-processing sometimes very expensive, they are using many chemicals, and this kind of sample handling, sometimes we need the very large volume of the samples, they are collecting very large volume of the blood samples, and also it is very time consuming process, because some trained personnel they need, they will handle your samples, and they will use this kind of expensive instruments, and they will incubate your samples, and it may take longer time, around more than sometime few hours. That's why you have to wait maybe at least one day sometime to get your diagnostic report. Sometimes it is very urgent, because you wanted to see that value, this diagnostic value, like within a minute, because you have to, in a critical condition, just think about ICU in the critical conditions, you have to know, you should know this value immediately, but this diagnostic method is taking too much time. It is something difficult to save the patient life, that is why. And always we need the trained personnel, and complex setup we are using, and there is a lack of miniaturizations, because this is the bulky instrument, you cannot use near the patient. So, we need some special setup, and no on site, this kind of setting we cannot bring on site near field testing. So, this is the problem we have. So, we need to solve all the problems. So, that how we can solve that I will discuss. That problem we can solve using one very important tool, that is called biosensors. So, then what is the biosensors? So, biosensors is a analytical device with one or more biologic recognition element. Just see, biologic recognition element we are using, there is very specific biological recognition element we are using, that can detect the certain target analyte of interest. So, biosensor is a technology that can help to detect some certain molecules, and we can easily we can make very small device and user friendly device by using the biosensor technology. So, I just last slide I discussed so many problems, that all the problems can be addressed, can be solved by using this technology. So, let us come. So, biosensor, I told you different kind of bio recognition element generally we are using the biosensor. What is this bio recognition element? Like antibody, like peptide, like aptamer, enzyme, DNA, RNA, this kind of bio recognition element we can use. So, you know the antibody, they are very much specific. If you have some disease like malaria, so we can use some malaria specific antibody, and that can target on your malaria antigen only in your sample, and we can detect those. So, this is called the bioreceptor or biorecognition element. They will capture your disease marker. So, I will come slowly one by one, but in my whole course, so not all, see, they all are the recognition element, but for example, mainly I will use the antibody. So, this antibody I will use just for example, this receptor on the biosensor surface, but anything you can immobilize on the sensor surface to detect the disease. Okay. See here in the right side, the application of the biosensor just I covered. It can be useful for the disease diagnosis, water monitoring, toxin detection, drug discovery. It can be useful for environmental monitoring. So, there is lots of applications we have in the biosensor. Now, come to the left side in this slide, you can see for the development of biosensors, some schematic we have drawn here. So, we need some part development for these biosensors. One is the transducers. So, transducers, just a surface, it can transform your one energy like chemical energy to the output energy like the electrical energy. So, on this transducer, in my biosensor part, I will teach you transducer just a electrode surface, because I will cover mainly the electrochemical biosensor here. What is electrochemical biosensor? Just I am coming. So, transducer is just a surface where we will immobilize this kind of biorecognition, this all the kind of biorecognition element we will immobilize on the transducer surface. Then we will drop the sample, and this sample will come to the antibodies or peptide, this bioreceptor on the surface. Then it will be I can say that absorbed on the surface, means they will target your biomarker. Then we can apply some chemical here, and that chemical reactions we can convert to a signal, and like the in the form of current, in the form of some charge we can see in the display. So, that current or charge we can amplify, there is some processor amplifier, then finally we need display like this mobile phone, there is a display, here you can see the data, some value So, there is different kind of biosensors available, kind of biosensor like electrochemical biosensor, colorimetric biosensor, mass biosensor, thermometric biosensors. So, electrochemical means there is a change of the electrochemical signal, means we can may use some chemicals on the transducers in this surface, then it will convert that signal to the electrochemical output like charge or current. Colorimetric means color change. Mass biosensors means there is change of mass on the sensor surface, and thermometric biosensors means there is change of the temperature. So, there is lots of this kind of technology we can use, but among those all the technology we will use the electrochemical biosensor mainly. So, why mainly electrochemical part I will cover here, because it is very simple technique and it is sensitive technique, it is we can easily miniaturize and it is very affordable technique. That is why mainly I will cover the electrochemical part. And also we can develop like smartphone based platform, so that we do not need any specialized any persons, we do not need any specialized equipment, no need any local reagent, and we can go for multiple detections. So, this is the main driving force of this course, that we can bring the rapid test method convenient immediate near proximity to the patients, right. Okay. So, this is the some references in this introductions part you can check, and in the next class then I will cover the lab on a chip or lab on a PCB technology that will be very much useful to bring the all the lab setup that I taught you on a chip, that I will cover in the next class. Thanks. Thank you all.